Спасибо, Нина. Guys, oh, my dears, oh, Chandrasekhar, please. Good morning, guys. That's some misunderstanding in the educational department. So, and at that time, 8 o'clock, there is no timetable in the technical department for our lecture. So, but I will deliver it right now. Maybe next time they will change the room number. But by now, please listen because this is important lecture for HIV. And Chandrasekhar and Goldheart, please spread or distribute this information about the link of the lecture and room. Okay? Thank you very much. So, and please, we start our lecture with HIV. Oh, Vishesh Shukla is already there. Good morning, everyone, guys. Please inform your group mates and everyone that we start now. So, today is the HIV. And of course, this, oh, thank you very much, guys. This is the topic is of utmost importance because it is HIV. And I would like to start with the statistic. First, in the first portion of our statistic, I would like to give you these figures. So to understand the morbidity and mortality within these almost more than 30 years, because you know that first this disease had been, the symptoms of this disease had been found in 1981, and the microbe had been isolated in 1983. So from 1983, the global statistic is considered on HIV morbidity and mortality. Morbidity, it means the number of cases, and mortality is the number of deaths. So then, here we have summarized statistic, and from here you can see that 75.7 million people have become infected in between 1983 till 2019. Within the same period, 32.7 million people have died from AIDS. And on 2019, it was considered that 38 million people live with HIV. And in 2019, around 1.9 million people become newly infected with HIV. And here you see that the number of annual cases drops a bit because it was 2.1 million in 2015. And within the 2019, 690,000 died from AIDS. And again, it is much less than in 2016, for example. Within 2019, newly HIV infected cases makes 1.7 million. And among children, and that's, please pay attention, that the number of cases drops dramatically, especially among children, because a lot of programs or several programs of different foundations are working hard to reduce the prevalence of the disease, especially in newborn babies and in children, because it was 300,000 in 2010 and it drops twice in 2019 down to 150,000. Then so, a fresh statistic. And I have to tell you that because we are talking about global statistic, it is difficult to get statistic the year we are here. So, for example, it is no global statistic exists for 2021. So it will be considered and calculated by the end of the 2021, and most probably it will be available somewhere in the middle of 2022. So that's why now we have statistic for 2020. And you see that now approximately 37.6 million people live with HIV. And of those, 35.9 million were or are adults, and 1.7 million are children under 15 years old. And around 690,000 people died from AIDS-related illness worldwide in 2020, and compared to the, as we said, 1.2 millions in 2010. So the situation is a bit better right now. But please take a look at this slide. That's something about the bad habits and some disorders that may increase the risk of the HIV infection. And you can see here that the risk of acquiring HIV is 29 times higher among people who inject drugs and 26 times higher among gay that have sex with men. Okay, right? So for those who have sex with men that have sex with men. So then 30 times higher for professional sex workers and 13 times higher for transgender people. So this is something about the very important epidemiological features of the disease, because here you can see now how most probably the HIV spread, and that is worldwide. Now let's take a look at the rotaviruses themselves. You're supposed to know that rotaviruses supposed to have common characteristics, and the unique feature of rotaviruses had been retroviruses, sorry, had been found far ago in 1970, like you see here. So retroviruses are enveloped positive strand RNA viruses with a unique morphology and means of replication. And they are really absolutely different. So that's why they belong to the last type 7 according to Baltimore virus classification. The most unique property is that they encode an RNA-dependent RNA polymerase called reverse transcriptase. So these RNA viruses replicate through a DNA intermediate, and the DNA copy of the viral genome is produced by RT. But then what is important that the DNA copy is not single-stranded but double-stranded. So they make complete copy of genomic RNA. And this complete copy of genetic RNA may be integrated into the host chromosome. But my dears, that had been found by Termin and Baltimore in 1970. They found this unique activity of the retrovirus specific enzyme. Now we take a look at the retroviruses classification. The family Retroviridae can be classified into two large groups or subfamilies, orthoretrovirinae and spumaretrovirinae, right? And in spumaretrovirinae, we have just spuma virus, and we have monkey simian formivirus and human simian formivirus. Then we have so many genera: alpha, beta, gamma, delta, epsilon retrovirus. And the last one, that is lentivirus, where human immunodeficiency virus belongs to. So family or genus lentivirus from the subfamily Orthoretrovirinae. I won't mention all others, but I would like to underline the first Rao sarcoma virus. That's very important virus of the poultry. So Dr. Rouse in 1905 found that something or some virus, in fact, poultry and cause the malignant transformation of the cells that was called Rao sarcoma virus. That was first retrovirus discovered. Okay, very few words about the rest of the retroviruses except for HIV, and then we concentrate on HIV only. So you can see here three subfamilies of the retroviruses, and you can see them. So a few words about each group. Spumavirinae. By now we don't know whether they are able to cause clinical disease or not. But what we know is that they produce characteristic vacuolization of the cells, so they may cause so-called foamy cytopathological effect. So it is called human formivirus. Then the group 4 is endogenous viruses. What does it mean? Endogenous. What indication do we have in the word endogenous? That means that this virus or the particular base pair sequences of the DNA are introduced and irreversibly, we think, integrated into the human genome. And these viruses make around 1% of the human chromosome. So we think they are ultimate parasites and they are usually transmitted vertically from generation to generation. We don't know much about this endogenous virus and we don't know the importance of them and the human pathology by now. Then, one more group of retroviruses is so-called oncornaviruses. They are those that can cause cancer. So we call them oncogenic retroviruses. So there are many animal oncogenic retroviruses, but human are HTLV, human T lymphotropic viruses 1, 2, and 5. They are called RNA tumor viruses. They cause immortalization of the transformed target cells. We believe that many of oncoviruses express viral oncogenes, analogs of the cellular growth controlling genes. Later on, we would have a lecture, separate lecture for on coronaviruses. So we discuss more about those viruses. By now, you can see that retroviruses can be classified according to the structure and symmetry of the uh, core. And that's important that, for example, on coronaviruses have C type of the core. C, this is uh, geometrical icosahedral structure, while the D type, this is the structural type, is when the core is cylindrical and appears truncated cone. So that is typical, type D is typical for HIV viruses. So it is very well recognized under the electron microscope according to its structure, to the shape and the symmetry of the nucleocapsid. I would like to remind you that in enveloped viruses, they have envelope with surface glycoproteins and nucleocapsid. That is a nucleocapsid with the uh, Mm, or in HIV and some other viruses, it is called core because it is very complex with RNA with the genome inside. Okay, so and the last subfamily is so-called lentivirinae, and these are non-oncogenic, my dears. They are non-oncogenic viruses, but they are cytocidal virus or those that can cause the death of the cells. And they are slow viruses associated with neurologic and immunosuppressive disease. So they cause slowly progressive chronic diseases, and one of the most common is AIDS. And AIDS stands for Acquired Immunodeficiency Syndrome, was primarily described as a disease of homosexual men, Haitians, heroin addict, and hemophiliac 4-H club. So it was found that the people from these communities start dying of normally benign opportunistic infections. And the very first syndrome had been discovered in 1981 and was pneumocystis carinea pneumonia, unusual in any other groups of individuals. So when the virus had been discovered, it means they had been isolated in culture. On May 1983, by Montagnier in Paris, the Institute of Louis Pasteur, and on that time he published a description of a known virus and the first name of it was LAI. And in 1984, Galore in National Institute of Health in the United States reported the isolation of also new human virus, and the name had been given HIV-1 from patients with lymphadenopathy and AIDS. So the syndrome had been discovered and named before the virus had been isolated. Later on, it appeared that those two viruses were the same and that's why they combine the priority, and Gallo and Montagnier shared the priority of discovering of HIV. Now, what do we know about the virus? It is spherical, yes, and as I said, it is enveloped virion, and it is of middle size from 80 to 120 nanometers in diameter. And you can see very well, and the whole world knows this virus by the D-type core, is a cylindrical capsid surrounded by the envelope containing viral glycoproteins. The core morphology, as I said, is used to classify the genus of the virus. It resembles a truncated corn. So, few words about the viral genome. It is single-stranded RNA positive sense molecule from 9 to 10 kilobases in length. But then you can see here very well that it is not positive and negative strand, but it is the genome of the HIV is diploid. We have two absolutely identical copies of RNA genome. Inside the core, we may see the so-called reverse transcriptase or RNA-dependent DNA polymerase. And also there is integrase there, one more enzyme, and usually we have very few copies of reverse transcriptase, but 10 to 50 copies per virion of the integrase. Then you can see here also that two cellular tRNAs that supposed to be used as primers for reverse transcriptase are base paired to each copy of the viral genome RNA. So I underline that it is cellular transport RNAs picked up from the previous host cell. And also the virus-encoded protease is required for production of infectious virion. So these three enzymes, reverse transcriptase, integrase, and virus-encoded protease, are very important targets for antiretroviral therapy. So you can see here the structure of the genome, and you can see that in this genome, we have quite important sequences. They are called LTR sequences. So long terminal repeat sequences that contain very important sequences, promoters and enhancers and other genes, sequences that are used for binding various cellular transcriptional factors, and that is for regulation of the integration of the virus into the host cell chromosome, and also the important in the regulation of the genome transcription. And you can see here that we have three structural genes, GAG, POL, and ENV. Can you see here? And the rest 7 to 8 are accessory genes. So I would like to remind you, what does it mean? Structural genes and accessory genes. The structural genes, when they are expressed, it means they are transcribed and translated interpolyproteins that then will be cleaved, but then those proteins give rise to the virion themselves. So structural proteins are those that will be enclosed into the virion. After the proteolytic cleavage of the polypeptides produced out of the expression of these three genes. And you can see here how proteolytical cleavage with virus specific protease will occur of the GAG gene product and this one Pol gene product and ENV gene product. And GAG means group specific antigens. What does it mean, group specific? They are conservative antigens that are not keep changing very quickly. And usually they are hidden inside the virion. So these are the proteins coding for the genes that code for core proteins and structural proteins. P24, see here, this is a major core protein. Then P17, that is the matrix protein. And P7, this is the nucleocapsid protein. P7 here or P9 in some other HIV viruses. So, nucleocapsid protein, see core nucleocapsid and matrix protein. They are group-specific antigen. Then we take a look at the pol. Pol gives rise, pol, name of the gene, uh, is due to the polymerase. So, and the polymerase, it means this gene codes for three very important enzymes. Reverse transcriptase with the molecular weight, this is the numbers, these are the molecular weights. And the molecular weight of the reverse transcriptase is P6651. Then integrase P32 and protease P10. And ENV is the envelope protein, envelope gene that code for the envelope glycoproteins, and we have two. We have transmembrane GP41 protein and surface structural unit, that is GP120 or surface protein. So we will discuss, yeah, I think you understand the function of the structural protein because we have studied already several types of the viruses, right? So, and the surface glycoproteins mediate the attachment and adhesion and entry of the virus. And the M protein is responsible for interaction between envelope proteins and core proteins and important in morphogenesis. Then the core protein is important in the protection of the nucleic acid, and so do the nucleocapsid protein, important for this. And the enzymes mediate the replication cycle. So except for the structural genes, we also may see several types of the accessory, so-called accessory genes. They all have different and multiple functions. Out of all those, the most important are TAT and REF. TAT is the transactivator of transcription. And it is important. Why do we call it transactivated? Because it is able to activate the other viral genes expression, but also some cellular genes transcription and translation. And also RAF is important, that is P19, that mediate the regulation of RNA splicing, because this process occurs within the replication cycle. And the product of this gene promote the export of the RNA from the nucleus into the cytoplasm for translation of other proteins. And also some other accessory genes are important because their products regulate usually the life cycle of the virus, because the virus may occur in the chronicle latent stage, but also some genes, for example, NAP gene, may activate the progression of AIDS by activating of the uh, rested T cells, differentiation and proliferation. So the VPU product usually facilitate the release of the virus. So it means the formation of the infectious virus particles. So it is responsible also for the decrease uh, of the CD4 numbers on the cell surface. Few words about the replication of the retroviruses. Uh, you can see here that this is T helper CD4 positive lymphocyte with the CD4 molecule, and this is the HIV with GP120, and this glycoprotein has lollipop structure. So this is lollipop made up of GP41, and the candy up on the top is GP120. So, and the spikes, the glycoproteins, form the trimer spikes on the surface of the virion. And the function of these two types of glycoproteins will be different. The GP120 polymeric structure is responsible for tissue tropism, so it coincides or it matches with the structure of CD4 molecule, while the stock that is made up of GP41 function is fusion of the viral envelope with the cell membrane. So the the first step of the replication is binding. So, and you can see here that native HIV glycoproteins trimer forms uh, the spike, and by 100 GP120, the spike binds with CD4 receptor. But now we know already that this binding is not enough to infect the cell. The GP120 supposed to bind not already with CD4, but also a molecule that is called chemokine core receptor. And we have two types of these core receptors on the different types of the cells. The macrophages usually have G protein coupled chemokine receptor. This is a CCR5. And T cells usually have chemokine receptor CXCR4 on the surface of the T cells. So then this GP120 is supposed to bind with this. The only then mm, the GP41 molecule, that one, will be exposed to the cell membrane and mediate the fusion and entry of the virus into the host cell. And the envelope will be left on the surface of the cell. Here you can see this process, right? So receptor attachment to the CD4 molecule and the chemokine receptor, and only then the GP141 will be expressed. So the result is that the core, together with some viral proteins, will be released into the cytoplasm of the human cell. And then this chemokine, sorry, this receptor, or this capsid or core will be transported directly through the nuclear pores into the nucleus. Then in the nucleus, we have pre-integration complex formation. When we have the activity of the reverse transcriptase to produce DNA onto the on the RNA as a template. And then the double copy of the DNA will be produced, and with the help of integrase, the integration of the double-stranded copy will occur into the chromosome. So then usually we have the stage of latency when these viral uh, uh, the copy of the viral RNA, double-stranded DNA copy, will occur inside the host cell for a while. So for what while, it depends very much on the type of the virus, type of the immunity of the person, and so on. Then we have the activation of the transcription of the viral genome and splicing of the RNA. So they will be transported, and that will be the export of the spliced mRNA molecule from the nucleus into the cytoplasm, where the uh, translation of the spliced RNA molecules will occur into some non-structural, as you see here, VPU and F proteins, and also structural proteins. You can see that from the RNA that code for the ENV gene, the GP160 will be produced, then it will be cleaved into two subunits, GP120 and GP41. They will be glycosylated and processed, and then they will be transported to the cell membrane and expressed on the surface of the cell membrane. Then the core proteins also will be produced, and first as the some more RNAs will be produced, the GAG pole uh, polypeptide will be produced, then it will be cleaved in the CAG and pole separately. And the GAG proteins will be also sent towards the cell membrane, and the M protein will line from inside the inner layer of the cellular membrane, and then the P24 will be produced here, not completely uh, mm, not completely cleaved by proteases. That's not P24, but then there's a complex over the GAG proteins here. And then the export of genomic RNA produced in the nucleus will be released, and these genomic RNAs will be transported and taken to the cell membrane and enclosed into the uh, newly forming virus. And then as soon as this happens, the virus will bud from the cell membrane, and the final maturation of the virus. What is it? Final maturation. The cleavage of the gag polypeptide interstructural components will be released. So you can see here that the virions freshly released from the host cell, they don't have this typical shape of the truncated core. Then this will appear after a while when the final maturation occurs and all gag products will be proteolytically cleaved into active uh, group-specific proteins. Then as soon as we see the truncated core, it means this virion is infectious already. So it may enter a new cycle and cause the infection of the new cells. So here are some details of the translation. As we said, all structural components, all structural genes are translated as large, long polyproteins. The gag pole, and envelope are mRNAs. And those polyproteins then need to be cleaved here, right? So without cleavage, the effective active proteins will not be produced. And then self-assembly is really very difficult or very complex, I would say. It is processed in the endoplasmic. The glycoproteins are processed, of course, in the EPR reticulum and Golgi apparatus because they need to be glycosylated and converted. So then they will be exposed here, and you can see here GP120 and GP41, right? Then the gag products partially cleaved will line from inside the membrane. Then the not completely cleaved gag proteins will bind with M protein, and the genomic RNA will be placed here. And then the process of the bonding occurs, and you can see here that is bonding, how it appears under the microscope. And you can see here the first immature virions will bond of the cell, and then they will be matured into the virions with truncated cone. See, initially we can't see this truncated cone there. The in release, the association of two copies of RNA genome and cellular tRNA molecules as themselves promote the development of the envelopment and bonding of the virion. And after the release, the complete cleavage of the gag-pull proteins occur, and the reverse transcriptase will be released, and the core is formed, so that we have infectious mature virus particles. You can see in detail the process of maturation, how the structural changes occur in the non-mature and mature virion. See, this is immature and this is a mature virion. Okay, so few words about the CPE effect typical for this virus. So this is syncytial formation. So that's important in understanding of this virus pathogenesis, because the very important feature of this virus is the ability to spread directly from infected cell to non-infected cell, and we can see the process of it. And as a result of it, the multinucleated giant cells will be produced with the that is the result of the fusion of up to 100 infected cells. And of course, this process over the syncytial formation enhance the cytolytic activity of the HIV, but then this process of syncytial formation occurs in the T cells only, but not in the macrophages, my dears. So this virus generally may infect two types of cells: CD4 positive lymphocytes and macrophages. My dears, in macrophages, the virus behaves itself absolutely different. It causes rather latent infection than the productive infection, and there will be no that high cytolytic activity in the macrophages. Few words about the really headache over the doctors and researchers they deal with HIV. Uh, this is genetic divergence, and the divergent population of viral genomes comprises a spectrum of not identical but related HIV genomes. What does it mean, my dears? It means that when it is called wild type of the virus, infect a new host, within a month, we may have in the host the diverse population of the virions, and they will be slightly different in the genome structure, and of course, it results in the difference in the antigenic structure, especially of the surface glycoproteins. Usually, we have one major type, and around 3 to 5 or 10 or some more minor types of the virions. Why it is important, I would like to say in advance. It is important because when we arrange a therapy for this individual, so usually we adjust therapy, and the therapies or drugs will down-regulate the replication of the major type, right? But then the minor types of the virus may escape this therapy. So with a particular type, when the major type will of the virus will be depressed and the replication of this type will be inhibited, then the minor types of the viruses start to multiply. So, and usually one of those that have beta mutations may start to be a major type. But then by these, the viruses escape from the therapy. That is the problem. So, divergent population of HIV genomes are found in all infected individuals. And of course, it is understandable that the highest divergence uh, is attributed to the envelope genes. So the higher antigenic divergence is typical for envelope glycoprotein GP120 and GP41Y because they are under antibody immunity press, biological. Uh, this is a sort of biological evolution if the neutralizing antibody match with the surface antigen. So the virus will be eliminated from the blood with the help of neutralizing antibodies. Then those mutations in the genes that result usually in changing of the envelope structure that allow them to escape neutralizing antibody activity will be selected, those types, so they may freely multiply in the human body. That is the very important consideration. And it is explanation why until now we don't have any effective vaccine. So my dears, that is the truth. We have very good treatment and we spend some time today for discussing all this. So, but uh, then the vaccination is still not present, really not developed. Okay, so go ahead with the pathogenesis of the virus. So we stated already that HIV has very strong tropism for CD4 expressing T cells, and usually this type of HIV is called T-tropic virus. But initially, when it enters the cell, usually it has tropism for macrophages, so it is called M-tropic virus. And then the HIV virus kills nobody. It's not that like this. So it is not a killer. So, but then it induces just immunosuppression. Then immunosuppression, this is a predisposing factor for other types of infections that kill the patients with AIDS. So the immunosuppression results from the reduction in the number of CD4 positive cells, followed by the of the it interferes with the helper function of T helper cells and the dysregulation of the immune response and delay type hypersensitivity reaction. That is important protective reaction in many types of infections, especially by protozoa or by uh, intracellular bacteria and some others. So, few words about the innate immunity cells, the role of the innate immunity cells in HIV infection. You can see here that HIV virus usually is transmitted through the enter through the epithelium cells, right? So, and the dendritic cells usually bind with the HIV and uh, with inside the dendritic cells, it may migrate to the lymph nodes, and dendritic cells usually transfer the virus to lymph nodes and introduce this virus to the T helper cells. So, and then the infection of T cells occurs within this process. So, in the Langerhans cells, usually mediate the degradation of the viral, and then it occurs also as a result, it occurs the presentation of the viral antigens in by dendritic cells to the T lymphocytes. So usually macrophages that take up the HIV work as a very important reservoir in the body. So it means there's a different importance of dendritic cells and macrophages. Dendritic cells usually infect the T lymphocytes, but macrophages serve as the reservoir. Those innate immunity cells uh, they are very important in T cell infection and keeping T HIV virus inside the body in the latent stage. And they have a special TLR for HIV and also outside the cell, and see here inside the endosomes, and that's important for those who studied immunity already, but you didn't, if I'm not mistaken. Okay, So, pathogenesis, initial infection occurs in the macrophages. And usually these are entropic HIV strains, and they are directed towards CD4 and CCR5 cells. Then macrophage lineage cells become persistently infected, and this is the macrophage here. So, and then circulating macrophages, microglial cells of the brains, and any other cells of this lineage, and the dendritic cells also uh, spread the virus and contribute to the clinical manifestation of the infection. And the T cell tropic HIV strains involve later in the infection usually. And you can see here the infection of the T lymphocyte by T tropic virus. So this is a normal and infected lymphocyte. And see here the infected lymphocyte and a lot of budding virions out of the surface. So what happens with the T cells as soon as they are infected with the HIV virus? So the virus causes direct CPE effect, and it may kill the T cells by several mechanisms, not the only one. First, uh, the multiple copies of the complete double-stranded DNA molecules. This is a complete DNA copy of viral genomic RNA are produced. Very few of them may be integrated with the host chromosome, but the rest non-integrated DNA molecules may occur in the nucleus, and so the once they will be accumulated in the nucleus, they disarrange the normal function in the nucleus. Then the budding over the cell membrane increase the permeability of the plasma membrane of T helper cells, so that also may help in osmotic lysis. Then syncytial formation is also one of the ways how virus causes the T cell destruction. Then the viral proteins can activate the processes of apoptosis of the infected cells. But these are just the direct action of the virus on the T cells. Uh, and the ability of the virus to kill the T cells depends on the amount of CD4 molecules expressed by the cell. So resting cells also can be infected, but the infection may be not that cytolytic in them. So it means if the T cells are activated with vaccination or with any other infections, and T cells stop to be resting cells and start to be differentiated and proliferating cells, then it will activate the or trigger the activation of the virus inside the resting T cell. So that's why there is a problem in vaccination of HIV infected individuals. We believe that by vaccination against any of the infectious disease, the vaccination may trigger the activation of the HIV harbored or infected T helper cells. So that's why it is really a problem. So, pathogenic mechanisms of HIV infection. You can see here the simplified scheme. So, when macrophages are infected, they persist inside the macrophages, and with the macrophagia, they may spread within the body, including the brains, where they may cause psychiatric manifestation over the HIV of AIDS that is called HIV-related delirium. Then, when T lymphocytes are infected, they may also cause persistent infection in the resting cells, but then they cause productive infection when the number of CD4 cells increase on the surface of the T cells when they activate. Then it leads to immunosuppression by many ways of killing T lymphocytes. And while T helper cells are killed and the number of them drop dramatically, then it opens the gates to all other so-called opportunistic infections, but also to the tumors. Why? Because T helper cells are the clue cells in regulation of not only antibody dependent but also cell mediated immune response. And also cell mediated immune response is important against tumor cells. So that's why the degradation of T helper cells also influence the ability of the body to stand neoplastic processes. Regarding the spreading by macrophage, this is a computer tomography of the brains in the patient with AIDS. This is a normal scan, CT scan, and this is a scan from the same patient 4 months later when he presented with the mental slowing and confusion. Profound loss of brain matter is seen, brain substance is seen, and enlargement of the fluid field ventricles occur. So that how it is seen histologically. So the cause of the disease. Usually the cause of the disease parallels with the CD4 T cell numbers and the amount of virus in the blood. So here with the green color, we can see the amount of the virus or each P24 antigen in the blood. And the red one is number of T helper cells, and the blue one is the antibodies against HIV. So, and initially, HIV infection is associated with the burst of uh, virus reproduction in the blood. So we can see the very high peak of the viremia. Viremia is the presence of the virus in the blood, right? So, and they appear very sharp and then they disappear from the blood. My dears, why they disappear from the blood? See, when the antibodies rise, the antibodies act as a factors that eliminate the virus from the blood. So within this time, usually that is called acute infection. The so-called mononucleosis-like syndrome may occur. Uh, Why do we call it mononucleosis-like syndrome? Because initial symptoms may be similar with that caused by cytomegalovirus and Epstein-Barr virus. So with the uh, temperature and some signs of the inflammation and so on. So then, uh, when virus level or viral load in the blood decrease, when antibodies eliminated, then within this time the virus will occur in the lymph nodes. So it may persist there and it shows the so-called latent period or period of chronic lymphadenopathy. So we don't see very clear symptoms of the disease, while we may see that the lymph nodes start to be enlarged in the body, fluctuant, so then it is a sign of inflammatory response in the lymph nodes. Then when CD4 levels, initially they don't drop that quickly, but then when they drop, the structure of the lymph nodes is destroyed, and then within that period, the patients start to be immunosuppressed. So when they start to be immunosuppressed, so then first subclinical uh, uh, dysfunctions may occur, and then the number of skin and mucus infections may occur, and so that will be the result of the immunosuppression. So we can see a large copy of the same. So, and after the stage of opportunistic infection, the systemic immune deficiency occurs when the number of the CD4 cells drop to less than 200 in 1 ml. There's the larger copy. See, this is CD4 numbers drop below 200 per uh, millimeter, cubic millimeter. A reduction in number of the CD4 positive cells may result from direct activity of the virus, right? Do you remember we mentioned? But also, do you know that T cytotoxic lymphocytes may identify infected T helper cells, so they will also cause killing of these infected T helper cell. That is called cytotoxic T cell immune cytolysis. So it will be a war between T cytotoxic lymphocytes and T helper cells. So then natural terminal differentiation of T cells may be affected due to the response to the large number of HIV antigens challenge. So then it will be like T cell anergy and the increased release of the virus into the blood as the number of CD4 cells decrease correlates with the development of the symptoms of AIDS. Yeah, this is more for those who studied. I won't stop on it, who studied immunity. So you just take a look at it if you would like to know more. Then immunopathology. Neutralizing antibodies that are directed against surface GP120 may participate also in antibody-dependent cellular toxicity because the antibody-covered HIV may be eradicated by macrophages and NK cells. But antibody-coated virus is infectious if it is neutralized by antibodies, so it can be uptaken by macrophages. CD8 T cells generate response against HIV-infected cells and also produce suppressive factors that restrict viral replication. They promote very important direct cytotoxic activity. And this activity reduces the number of HIV-infected cells. And this is very important. So, but the immune escape is very typical for HIV, and it is resulted from the ability of HIV to incapacitate the immune system, persist in the macrophages and lymphocytes, and alter its antigenicity. Few words about the epidemiology of the process. We believe that HIV is derived from the simian lentiviruses because the variant of the HIV that is HIV-2 is very similar with the simian sooty mango-based virus in West Africa. And by the way, HIV-2 is the type of the HIV virus that's more typical in Africa. The initial human infections may have occurred in Africa because in the blood banks there from 1960s the HIV virus had been discovered. So then a lot of people migrate that time in the mid and late 60s from Africa to the rest of the world. So that's how we think the virus had been spread in worldwide. So the transmission of the virus occurs through blood because HIV is present in the blood, through semen, vaginal secretion of the infected individuals. So the virus usually spread through sexual contact and exposure to contaminated blood and blood products. It can be also transmitted vertically from mother to child. You know what population is at risk. Take a look. We mentioned already those who are at higher risk of getting the disease. So, few words about the clinical symptoms of the disease. As we said, the initial acute infection may resemble mononucleosis or influenza-like symptoms. And these symptoms usually subside within 2 to 3 weeks. Sometimes rash may occur, or septic meningitis symptoms may occur 2 to 3 months after infection. These symptoms stem from immune responses against HIV. The initial symptomatic period of HIV infection is followed by a period of asymptomatic infection with generalized lymphadenopathy. This is it. And then it depends on the type of the virus and type of the patient, because this chronic period or persistent period may occur from half a year, may last from half a year, or years, or even decade. So then these syndromes occur over the AIDS, right? And they will be formed. And it usually starts with skin and mucous membranes infections and the defects. And that is just a result of the deterioration of the immune response. The onset of these opportunistic infections occurs and correlate with the reduction of the CD4. When it drops below the 450 somewhere here, so that means that the stage of opportunistic infections occur. And then the AIDS occurs when the CD4 drops below 200 in ml. It involves the onset of more significant diseases. These the important are HIV wasting syndrome, when we have weight loss and many month diarrhea, Kaposi sarcoma, this is indicated as disease, pneumocystis carinii pneumonia, that's typical for AIDS patients, and TB gotten from not highly pathogenic uh, species of mycobacteria, but from, for example, mycobacteria avium intracellularly. So this is example of wasting syndrome and lipidystrophy in AIDS patients. Then you can see here the aspergillosis. That's very typical marker disease in AIDS patients, where the aspergillomas are formed, and you can see them in the chest X-ray examination. Then candidiasis occurs, but not just oral or vaginal local candidiasis. So candida may get access to the blood and spread and cause generalized infection. So we may have candida sepsis, oral candidiasis, uh, esophagitis due to the candidiasis, trachea infection, bronchi, and lung infection, and quite often it is lethal in AIDS patients. Then disseminated cutaneous fungal infection. There's example of sporotrichosis in a patient with AIDS. And here's the uh, Kaposi sarcoma associated disease. When this sarcoma, usually it involves the mouth, lesions are usually located in the gum or hard palate and can appear macular, nodular, rise, or ulcerated with color ranging from red to purple. So, and oral hairy leukoplakia of the tongue usually occur, and this is also the neoplasia. So, and you can see here the example of Kaposi sarcoma. How do we perform lab diagnosis? Generally saying, the recent infection or late-stage disease can be indicated by the finding of P24. Do you remember this? See, we can find P24 initially or at the very late stage of the disease. But then we don't usually use it so often. The very important marker is appearance of the antibodies. See here, they appear quite early within disease and persist for a very long while of the uh, mm, during the almost all stages, except for the very last one when due to the immunodeficiency and anti-HIV antibodies wane from the blood. So, because anti-HIV antibodies occur within a very long while, so serological tests are usually tests of choice to document the HIV infection. But these tests supposed to be used in two stages. First, for routine screening, we use ELISA to find antibodies against HIV-1, HIV-2, but it may give false positive result. But this is a screening test that is developed so to find anything that looks like even the anti-HIV antibodies. Then the result of routine screening tests need to be confirmed by more specific tests. And usually we use Western blot analysis because it is more specific. And as a result of Western blot technique, we may demonstrate not the presence of any antibodies, but we may answer against what viral protein the antibodies are produced. Then this is the basis for diagnosis. Of course, reverse transcriptase is a very good tool, especially in the moment when the antibodies are not produced yet. Take a look at this. This is called seronegative window period, when the antibodies are not produced. If you are suspicious, we may take a look at the presence of the viral unique genes code for RNA polymerase in the blood. This is, and also it is the it is test where we can calculate the number of the viral load, the number of the copies in 1 ml of patient blood. So this is a very good tool also to control the effectiveness of the treatment, because that is the only test that may confirm whether the number of the viruses drop in the blood, in the patient blood, or stay high. Uh, Culture of the HIV can be performed, but it's not routinely used in the diagnosis. It's not that difficult because it grows very well in the patient's peripheral blood mononuclear cells that will be stimulated by interleukin and with polymorph peripheral blood mononuclear cells from the donors in the presence of IL-2, because it's a very good stimulator of T helper cells differentiation and proliferation. Usually, lymphoblastoid cell cultures can be used, and that is rather used for immunological studies. My dear, the number of T cell subsets cannot really be differential diagnostic marker because T cells may drop, number may drop within some other situations and within some other infections also. But then it can help to identify the stage of the disease. Few words about the evolution of antiretroviral therapy. First treatment had been done with zidovudine. It was released in 1987. And first group of drugs, and zidovudine belongs to, is nucleoside reverse transcriptase inhibitors. I hope you know what is this. This is the azidothymidine. This is the very first acting substance that mimics the nucleoside when it had been added to the during the transcription. So it will be a termination of the transcription. So the DNA copies will not be produced, so the abortive infection occurs. So second anti-HIV drug had been produced as non-nucleoside reverse transcriptase inhibitor. Then the protease inhibitors occurs and integrase inhibitors had been found. So a few words about this. So nucleoside analogs reverse transcriptase, you can see the very first one and then the analogs of it. So after incorporation of the nucleoside analog into DNA, the termination of the chain occurs. Then the second type of drug licensed for anti-HIV treatment had been proved in 1990s, that is protease inhibitors. They block the morphogenesis of the virion, inhibiting the cleavage. Do you remember the cleavage of the polypeptides, GAG and GAG-pol polyproteins? And here you can see the several names like saquinavir and nelfinavir and others of the protease inhibitors. Then the non-nucleoside reverse transcriptase inhibitors had been licensed and implemented in the mid and late 90s of the previous century. These drugs change the properties of the reverse transcriptase active site, and by this they reduce its activity and thereby inhibit HIV replication. You can see the last one was F5RNs and some more, the names are here. So those three types, my dears, three types, non-nucleoside analogs, protease inhibitors, and nucleoside analogs, they used to be combined to perform highly active antiretroviral treatment. Now it is called and well known as combinatorial antiretroviral treatment. So usually cocktail contain several antiviral drugs with different types of actions. Because it was, and now we still think that this cocktail prevent the early development of the antiviral drug resistance by the virus. Then some more types of the antiviral drugs, antiretroviral drugs, and be added. So, for example, fusion inhibitors. So, this is enfuvirtide. So, it prevents the HIV virus envelope from fusing to the host CD40 cell membrane, and so it blocks the infection. that was introduced, I think, in 2007. Then the second type, core receptor antagonist. We believe that core receptor binding is needed. See here, CCR5. So core receptor like maraviroc, ivcriviroc, they block the binding of GP120 with the core receptor, so it prevents also the entry of the virus into the cell. And then integrase strand transfer inhibitors. So briefly, INSTIS. Uh, these drugs such as raltegravir, LV-tegravir, and dolutegravir interact with the integrase enzyme produced by HIV particles, preventing it from inserting the viral DNA produced via reverse transcriptase into the genome of the host CD4. That's very important. So it prevents integration of the viral genome into the host cell. And the last group, very well used and known by now, is for the attachment inhibitors. We have now the drug based on fostemsavir, and the drug is called Rucobia. It is the attachment inhibitor, and this novel class of HIV drugs that bind with the GP120 protein on the outer surface of HIV, preventing from the attachment to the CD4 T positive lymphocytes. And with the help of Rucobia, that's one. So now the development of long-lasting therapies is known, and it is proved already in the United Kingdom and the USA. So that this when the drug is injected once a month or once in 2 months, and it gives very good result. And of course, the adjustment of this type of therapy is very convenient for those who had to take pills, the cocktail of pills every day. That's make the therapy more available and more soft for people who need this type of therapy. So you can see a picture of the HIV infection, and you can see here the targets for therapy. See? So these are core receptor inhibitors. This is no fusion occurs, fusion inhibitors. Then reverse transcriptase inhibitors, protease inhibitors. The stage of integration will not occur if you use integrate inhibitors. And then also protease inhibitors act here, but also here during the maturation of the virion. My dears, now we have some new types of treatment. So it is believed that HIV, when it remains latently integrated in the host CD4 T cells for a very long time, this stage of integration can be interrupted. Karpinski and other authors developed a recombinase. This is an enzyme that is called BRAC1 that site specifically recognize the specific sequence in viral genomic DNA integrated into the host cell, and they will cut out this DNA out of the host cell chromosome. These doctors showed that this recombinase BRAC1 can effectively and safely remove integrated HIV provirus from infected cell in vitro and vivo in humanized mice. So now they are thinking about and they are arranging the clinical trials in humans, because that is a very important stage. It will mean that the T cells can be freed from the integrated viral genome. That's important. So, yeah, that's we've seen already. Prevention and control, education, blood and blood product screening only. So, vaccine against HIV is not available. That's it, my dears. See you next time. So we're a bit over time, but it was delayed with the start. So thank you for coming. Goodbye, guys. See you in classes. LA, LA1. Okay.